Okay, good morning, ladies and gentlemen. Time to start today's lecture. Thank you. So what I want to do is to carry on exactly where I left off almost a week ago, last Tuesday, by talking about halophiles, the first main group of extremophiles I'm talking about in these lectures. So just a brief reminder of where we find halophiles. Essentially, these are very, what I've been describing as niche environments. That is, there is a percentage of the biosphere, a very small percentage, quite unique in terms of certain inland lakes like the Dead Sea or the Great Salt Lake. And they can be things that are human sort of activity driven. Sorry guys, can't have some of the people talking when I'm talking, it completely throws me, so thank you. So you have to have human mediated evaporation ponds for making salt. And then natural environments again, sort of microbial mats and also underground saline deposits. So we find these by human activity of mining for salt, but these are natural deposits, sometimes kilometres below the Earth's surface. Okay, so what I talked about mainly in Tuesday's lecture was one group of halophiles, the aerobic heterotrophic bacteria. So these are typically found in seawater. So many of these are sort of like Selenivibrio casticula, are found often in seawater, but they can survive in a lot more salt than seawater. So just to always keep in mind, when you think about salt, the equivalent sodium chloride concentration for seawater, including all the salts in seawater, is 0.5 to 0.6 molar salt. So if you keep that in mind, then obviously when you're up at 4 or 5 molar salt, you have 8-10 times the salt concentration of seawater. And another genus, Halomonas, is found very widely spread around the world in all sorts of salt lakes, in North America, in sort of the Middle East, in parts of India and China. All of these countries with salt lakes, you find Halomonas all around the world. And it's a very halotolerant organism. It will grow in very low concentrations of salt, but also grow up to about 4 molar salt. And the key thing I was talking about in the lecture on Tuesday was this idea that when we grow Halomonas in particular, or other organisms similar to Halomonas, in rich medium, they produce glycine betaine as a compatible solute. So remember I talked about glycine, betaine, glycerol, and ectwine as being the 3 main compatible solutes. When we then put Halomonas into a defined medium, just with the glucose and defined salts, then what we found then is that apparently there was no compatible solute. And that's because we couldn't detect ectwine. So if you remember from last Tuesday, ectwine is an unusual cyclic amino acid derivative. So it's non-hydrogen negative, the test we normally use for amino acids. So it took work on the NMR by a guy called Erwin Galinsky at the University of Bonn at that time to work out that there was actually ectwine was produced in the minimal medium. Okay, so what I want to talk about then for new stuff today is I want to say a little bit about Dunaliella. Now, Dunaliella is, anybody who knows me a little bit, has come across me in the lab, or anybody will know this is my favourite organism. I first started growing Dunaliella in my final year research project. So I've had some Dunaliella growing somewhere all the way from 1978 until now. It survived for a summer in my mum's windowsill and things like this. It survived a trip to Germany and back. And so I've always had Dunaliella growing somewhere in the lab. So for those who did MBV 6303, the cells as factories, we talked about Dunaliella there as an industrial microorganism. I really just want to concentrate in these sort of few slides and hand out with how Dunaliella actually copes with high salt. So obviously the two things do overlap, but if you like, this is a distinct, different sort of look, if you like, at Dunaliella, rather than from purely a sort of industrial point of view. So it's a photosynthetic eukaryotic green alga. In most of these aquatic saline environments, like the Great Salt Lake and many of the lakes elsewhere in the world, it's the primary producer. So what I mean by that is it takes light energy, CO2, makes biomass, and then other things in the salt lake can actually act as predators on the Dunaliella, and you can get a very simple food chain built up in the aquatic lakes, possibly going as high as invertebrates in terms of brine shrimps. So Dunaliella is the primary producer in these environments. And the key thing about Dunaliella that we discussed for all the undergraduate students in first year in the lab, in the first year lab, is that you can actually grow Dunaliella over a range of salinities, measure the amount of glycerol inside the cell, and you get a lovely straight line effect. So as you double the amount of salt in the medium, you double the amount of glycerol. And we now know that Dunaliella produces glycerol using this glycerol cycle. This has been very well worked out now over a number of years. Okay, so hopefully the vast majority have got handout 4 in front of you. This is it on the visualizer now. This is showing you the glycerol cycle that Dunaliella utilises. Now, you'll see a theme from last Tuesday's lecture as well, that these compatible solutes are usually produced by 2 or 3 enzyme reactions from central metabolism. A compatible solute has to produce very quickly if it's going to be effective, so there's not any way you could have a long chain of enzymatic reactions. So usually there's just 1 or 2 things have to be switched on to produce a compatible solute. So in this case, dihydroxyacetone phosphate is the central metabolite. This is part of the glycolysis cycle, and you can either get to this point by fixing CO2, or it's also possible that you could utilize the storage compound in the alga starch, produce the dihydroxyacetone phosphate. So if the cells get a really big increase in salt, you may find that the photosynthetic mechanism is actually inhibited. So in that case, you'd have to produce some DHAP from starch to get this glycerol cycle going. And then once they got equilibrium again, photosynthesis could resume. So what they do is they switch on this enzyme here, glycerol phosphate dehydrogenase, which converts dihydroxyacetone phosphate to glycerol phosphate. And this actually takes place in the chloroplast. So the other thing to note in this diagram is that the upper part is the chloroplast. This is the chloroplast membrane here. And then the lower part is the cytoplasm. So enzyme 1 is fully within the chloroplast. So it's a chloroplastic enzyme, and it produces glycerol phosphate. And then the second enzyme is glycerol phosphate phosphatase, so that knocks off the phosphate group and makes glycerol. And enzyme 2 is both in the chloroplast and in the cytoplasm. So you have to have glycerol in the chloroplast. If you remember back to pictures of Dunaliella, the chloroplast makes up about 50% of the cell volume. So you have to osmotically balance the chloroplast as well. So enzyme 2 is both in the chloroplast and in the cytoplasm. And to get some glycerol phosphate into the cytoplasm, there's a transporter across the chloroplast membrane that brings it into here, and glycerol is also produced in the cytoplasm of the cells. So just switching on 2 enzymes, you can immediately produce glycerol. So glycerol production starts within literally minutes of a salt increase, and you get the glycerol being produced over a matter of hours, usually to get back to equilibrium. Now, if the opposite happens, if the cells are suddenly subjected to a decrease in salt, then they'll do one of 2 things. If the decrease in salt is really dramatic, they'll actually eject the glycerol from the cell. This will be a shock response. It's also very wasteful for the cell to lose all this carbon But if it's a big enough decrease, they will actually throw the glycerol out. If it's a small decrease in salt, they can then just go through the metabolic process. They can switch on enzyme 3, which is dihydroxyacetone reductase, which converts glycerol to dihydroxyacetone. And they've got a kinase enzyme, dihydroxyacetone kinase, which will convert the DHA, the dihydroxyacetone, to dihydroxyacetone phosphate. These both take place in the cytoplasm only, and then the DHAP is then transported back into the chloroplast to go back round the cycle again if necessary. So there's only 4 enzymes involved in the glycerol cycle, 2 that switch the glycerol synthesis on, and 2 that degrade glycerol back to DHAP, and then it can actually then go back to starch and as a storage product. If the cells are then in low salinity for a significant length of time, then they'll just start to accumulate starch again. So it's a really neat system. One of the reasons I always stick with this is not just my bias towards Dunaliella, but this is really a very neatly worked out system that's been studied for many years now. These enzymes are all well characterized, their place in the cell is well characterized, and I think this cycle is essentially completely sorted out. You can see from the, I'm not going to show you that particular reference, but this is first done in 1992, so it's a long time ago. But all the subsequent experiments have actually proven this correct. And that's the great thing about science, is that we're always checking these things. So if somebody suddenly came up with something different, we'd have to look at it and see if we have to rejig the glycerol cycle, but so far it's stood the test of time. Okay, so what the glycerol cycle allows the cells to do is to produce sodium chloride, to produce glycerol in response to sodium chloride in the medium. And this is, I say, a linear response. I think I showed you that in the 6303. So that as you double the salt concentration, you double the amount of glycerol being produced. And again, I've taken this table from an even older paper, because again, this hasn't changed in many years. So this is the response of Dunaliella to increasing the salt concentration. So in this table, the left-hand column is just the amount of sodium chloride in the medium. So starting 0.5 more or less at seawater, doubling to 1, 2, 3, and up to 4 molar. The second column is showing the intracellular concentration of sodium. So it's negligible at the lowest level. It does go up a little bit, at 1, 2, and 3 molar, but only to sort of 19, 20 millimolar or so. And then when you get to 4 molar, it does go up to nearly 100 millimolar. So what that's telling you is that at 4 molar, you are starting to get towards the limit of the cells growing. They can go a bit above that, but that jump in sodium actually tells you that they're starting to, it is starting to stress them to keep the sodium completely outside the cell. The other intracellular cation, potassium, now we talked, and we will talk a bit more about potassium being a very benign cation, and certainly Dr. Baker will talk about this. And you can see that it remains about the same level in Dunaliella, somewhere around about 200 millimolar, a bit less, at lower salt concentration, a little bit more. But the osmotic change due to potassium is trivial, basically. It's not making much difference. It's probably there just to protect some of the enzymes. And then the final column is the glycerol column. And so at the beginning, you've got about the same amount of glycerol as salt, and then you've got about 50% more at 1 molar. You've got almost double then at 2, 3, and 4 molar. And that's correct, because if you think about it, sodium chloride switch into sodium and chloride ions. They are both osmotically active. Glycerol is one molecule. So the first approximation, you need double the amount of glycerol that you do salt. So why then in the low salt concentration is it only 0.5 molar? Because there is enough other things inside the cell at 0.5 molar that still do the balance. As you increase it, that becomes less and less, and so you have to increase the amount of glycerol relatively to the amount of sodium chloride. But the key take-home message is that it's produced in very large amounts, and it's a very effective and benign compatible solute, allowing Dunaliella to grow at least up to 4 molar salt, and probably with a bit reduced growth rate as high as 5 molar salt. Okay, so I did mention in the previous lecture that glycerol is the cheapest compatible solute to produce. It's cheaper than ectwine and glycine butane on the basis of the ATP equivalents required to produce the glycerol. So why is it only found then in halophilic eukaryotic microorganisms? Why aren't all the Halomonas and Selenovibrio and everybody else, why are they not using the cheapest compatible solute? Because when you do get a chance, if you look back to handout 2, then what you'll find is that none of the other organisms, apart from algae and a few fungi, actually utilise glycerol. Now I've actually got handout 2 here. So just to remind you, just at the bottom bit here in handout 2, these are the only organisms that will use glycerol, nothing else, all the way up that big table will use glycerol. The cyanobacteria do use glucosyl glycerol, they're getting somewhere close to it, but the only one that actually utilises glycerol are Dunaliella, a few other halophilic, halotolerant algae, and also halophilic yeasts, things such as Debromyces and other yeasts will use glycerol and other polyols. And essentially the answer to this question is it's a membrane problem. So effectively for most organisms, for most bacteria, if you add glycerol into the medium, it's very easily taken up, but it also is very easily leaks out of the cell. So if a E. coli cell or a Halomonas cell tried to produce several molar glycerol within the cell, even if you gave them the machinery to do that, in the enzyme machinery, what would happen is the glycerol would leak out. So you'd also have to change all the membrane architecture as well for glycerol to be an effective compatible solute. So glycerol is the best compatible solute energetically, but it has this problem of being permeable to most membranes. And the Dunaliella and the Debromyces and other fungi and algae have adapted their membrane structure to retain glycerol. And I'm amazed after all these years that I've been giving this lecture, we still don't actually know how that's done. It's been a sort of back project in my lab for literally decades. We've never really had a chance on funding to really look at it. And there's been a few ideas in the literature. It probably has something to do with the sterol sort of composition of the membrane. Things like cholesterol, but you do find these sterols in algae as well. It's not just in humans. And it's probably likely that there's some effect of the sterol composition that actually allows the fungi and Dunaliella to actually retain the glycerol. There's sort of circumstantial evidence that the sterol concentration is different in different types of sterols, but nobody's really written a paper convincingly that will back this up. But undoubtedly there is something there, because Dunaliella retains glycerol up to 8 molar within the cells. Debromyces, the very salt-tolerant yeast, does exactly the same, whereas no sort of prokaryotic cell or no cyanobacterium will actually be able to do this. Right, that's all I'm going to say about Dunaliella. It stands alone for all the students here in 340 and 6340. If you did also NBB 6303, it fits in with what we talked about in terms of Dunaliella being a very salt-tolerant organism. But as you know from those who did 303, glycerol is not one of the products from Dunaliella, but it is a very important part of the physiology of Dunaliella, and is absolutely required for it to grow in its environments. So the classic place to find Dunaliella is either in somewhere like the Great Salt Lake or the Dead Sea, but also in a rock pool. So the good thing with Dunaliella is it's a very adaptable organism, and we could sort of go across to Filey Brig or something like that, sample the rock pools, and we'd probably find some Dunaliella in these rock pools, even in the coast of the UK. And the reason for that is because it's good at responding to changes in the salt concentration. Right, so compatible solutes. So I've talked about things like Halomonas and Selenovibrio utilising ectwine and glycine butane. I've talked about Dunaliella and other eukaryotic organisms, algae and fungi, utilising glycerol. So what then is the mode of action of compatible solutes? So I'm going to give you a sort of a non-expert overview of this in this lecture, if you like, in the sense that I'm not a structural biologist, and then some of this will be taken up by Dr. Baker in his lectures when he's talking about how proteins are able to survive in high salt concentrations. So the key characteristic of a compatible solute is that it doesn't interact with proteins. So basically, if you've got 8 molar glycerol present inside a cell, it's actually not interacting with the proteins. It's actually forming sort of bounds around the proteins, allowing the proteins to carry on working in this very low water environment. But it's actually not being a denaturing compound. That's why it's compatible. And the word for this is a strong water structure former. That just basically means it binds lots of water molecules, and this allows it to keep a hydration shell excluded from the protein. So it covers itself in a hydration shell, and it then doesn't interact with the hydration shell of the protein. So this the way I tend to think of it as a non-structural biologist is they sort of bounce off each other rather than interacting with each other. So diagram 1 in handout 5, if you want to pull the other handout from today. I should also mention, just in case I forget at the end, I do have other copies of handouts 1, 2 and 3, so if anybody missed any of the previous handouts, then just come and grab them at the end of the lecture. Right, so that is my very, well, it's not my diagram, it is taken from a research paper, but a nice simplistic view of how compatible solutes work. So you just imagine a hypothetical globular protein that's shown in the diagram A in its normal conformation. So that is its active conformation where it's an enzyme. It's catalyzing its reaction quite happily. In B, it's actually been put into a solution of a denaturing compound, so something like urea. So urea is known as a very sort of strong denaturant. So if these little open circles in diagram B are urea, and what's happened is that the three-dimensional structure of the protein is broken down, and also that protein is no longer active either as a structural protein or as an enzyme. So if that was happening within the whole cell, then obviously cell metabolism would be breaking down completely. Now, what happens if you have it in the presence of the compatible solute, like a glycerol or a glycine betaine or ectoine, is that this hydration shell around the protein is formed. There's all hydration shells around the compatible solute, and so they just don't interact with each other. So instead of, you see in B, actually shows, the cartoon shows the urea being intimately acting with the protein, whereas in C, the cartoon shows you that the compatible solute doesn't interact with it. So that's, to be honest, quite a simplistic way of looking at it, but it's the way that I've certainly looked at it over the years. And it is correct, but I mean there is also more detail that you'll get from Dr. Baker in his part of the lectures. But that's what we're trying to do in this module for me, to set the scene for some of these things, and certainly for the halophiles and thermophiles, for Dr. Baker to give you more information on the structuring side of things. And it appears that all these compatible solutes work in effectively the same way. So whether it's a glycerol or an ectoine or a glycine betaine, they're all doing the same sort of strong water-forming structure and not interacting with the proteins within the cell. Now, of course, bear in mind that this is a simplified cartoon. There's lots of proteins in the cell. It's not just one, but this is just to demonstrate the key sort of relationship between the two. Okay, so moving on to a little bit of what the compatible solutes can do. I mean, I did mention glycerol in the 303 lectures, but ectoine and hydroxyectoine are also known to be very good at protecting cells, not just from high salt concentration, but also from high temperature and freeze-thaw cycles. Now, this is an important concept here, so that you'll find that compatible solutes are compatible against a number of different sort of stresses, not just high salt, certainly high temperature, and also freeze-thawing are. They can, in this same sort of way as you're seeing in the handout, they can actually protect the enzymes and the proteins against these stress effects. Okay, so the second part of handout 5 is just giving you two examples of the protective effect of ectoine. So the enzyme here is lactate dehydrogenase. It's just a commercial suspension of lactate dehydrogenase bought from Sigma Chemical Company of River. And what they've done here in this particular paper is they've subjected this lactate dehydrogenase to different stresses. So you can see that for the upper graph, this is temperature. So they start here at 25 degrees, and the different ones are either with no compatible solutes, so the open circles at the bottom line, there's no compatible solutes present. In the square one, that is betaine. The upper sort of triangle is ectoine, and hydroxyectoine is the top one. So I did show you on handout 1 right at the beginning of the lectures that there are two versions, if you like, of ectoine, one with the additional hydroxyl group. And it's very interesting to see in this particular experiment that it is actually significantly better than ectoine itself. So hydroxyectoine starting at 25 degrees, there's no effect on the activity of lactate dehydrogenase until about, certainly above 50 degrees centigrade. Whereas for the control, you can see it's coming down, so the time they get to 50 degrees centigrade, it's only working at about a third of its activity. And if you follow on, both for, you can see that the control then falls at 55 degrees centigrade. Ectoine also comes down here. Betaine carries on a bit further, and then ectoine here. But not worrying too much about the exact relationships, the key thing is that all of the compatible solutes significantly protect the enzyme in this key area, between 25 and 55 degrees centigrade. And also always bear in mind, and I'll emphasize this in tomorrow's lectures and thermophiles, that microbes cannot avoid temperature. If it's 55 degrees centigrade in the medium, then it's 55 degrees centigrade inside the cell. So it's really important that they have these protective effects. In the bottom diagram, this is just showing you the effect of freeze-thaw cycle. So again, the control is the clear column at the front. Betaine, ectoine, and hydroxyectoine are the three compatible solutes. So after one freeze-thaw cycle, then the control is dramatically less and is actually then disappearing away. Once you've done it 4 times, then basically it's all disappeared. Betaine is not particularly good at protecting against freeze-thaw. Interestingly enough, it's not only fractionally better than the control, but the back 2 columns, the ectoine and hydroxyectoine, show that even after 4 freeze-thaw cycles, you've still got at least 80 or 90% activity. Why is that important? Well, because if you've got an enzyme in the lab that you want to work with, you've got it stored in the freezer, you take it out of the freezer and you thaw it in an ice bucket so you can actually use it, and then you freeze it again, so that's one freeze-thaw cycle, and then a second one and a third one, and so on and so forth. So if you were not protecting it in some way, then after a few freeze-thaw cycles, you'd probably hardly get any active enzyme left. So it's really good to have something like ectoine and hydroxyectoine present to maintain the activity of your enzyme. Just to point out that why I think it does say hydroxyectoine. Right about from this paper was published, they're only just identifying hydroxyectoine, and sometimes it actually was just called Y. Yes, sometimes it is. So Y is hydroxyectoine. It was called Y until they actually worked out it was very similar to ectoine. Okay, so at the moment, just come back to the slides, when you buy your lactate dehydrogenase, other enzyme from Sigma or elsewhere, it will probably come as a suspension in sort of 3 molar ammonium sulfate. That's been the traditional way of keeping the enzyme active. And it's now becoming more common for ammonium sulfate to actually be replaced by ectoine, because ectoine is a much more friendly compound and will keep your enzyme active for much longer. So ammonium sulfate has been the traditional way of keeping enzymes sort of happy, but based on sort of results such as the one at the bottom of handout 5, you can see that it's actually a very good idea to utilise ectoine. Okay, so that's all I'll really say about compatible solutes, specifically about the mode of action. You'll get more information on this, I say, in the structural lectures that will fit in with what I've told you. I don't think anything I've told you is wrong, but it's certainly a fairly simplified view compared to what you can get from the structural sort of side of things. And what I'm going to go on to for the rest of today's lecture and finish off the halophiles is to go back to the other mechanism. So if you remember when I first started talking about halophiles, I said there was two mechanisms of growing in high salt. There's a mechanism we've been talking about now for quite a while, the low salting mechanism, where you produce a compatible solute. So now I want to go back to the other mechanism, which is the high salting mechanism, and look particularly at this group, the Halobacteriales, which have been really well studied for their particular mechanism of growing in high salt. And in particular, in the genus Halobacterium, it's an archaeon, so it's an archaea genus, and they require about 3 molar sodium chloride for optimum growth, and so they're irreversibly adapted to high salinity. So we've got a completely different situation right from the beginning. If you just grow Halobacterium in the lab and grow Dunaliella in the lab, you immediately see the difference between the organisms. Dunaliella is very flexible in terms of the salt concentration. Halobacterium requires a large amount of salt. And just a couple of years ago, I had an engineering student who wanted to work with Halobacterium, and she just couldn't believe the amount of salt she had to add to the medium. I mean, I think she just thought it was incredible. I mean, 3 molar sodium chloride, that's 175 grams of sodium chloride in 1 litre. So that's really a large amount of salt, a sort of 400 or 500 ml beaker, largely full of salt that you're pouring into 1 litre. And so it's a good thing, reason why when you do experiments, things really become clear. You realise just how much salt you're adding into these organisms, and they'll even take more than that quite happily. But the key thing here, leaving that aside, is that they are irreversibly adapted to high salt. So if you put Halobacterium into less than 1.5 molar sodium chloride, the cells literally fall apart. You can watch under the microscope as the cells literally disintegrate. And the reason for this is because the whole of a Halobacterium cell is adapted to high salt. So the cell walls, the cytoplasmic proteins, the ribosomes, they all have a high level of negative charges, mainly these CO- groups. And what this means is that if you don't give them lots of positive charges from the sodium in the medium, then they quite literally repel each other. Negative charges repel, as you know, of course, from long time ago in science, and then the cell just literally crumbles apart. So this is completely unlike the Halomonas or the Dunaliella, where the glycine beta-arrectoid in the Halomonas or the glycerol in the Dunaliella protect the cells without anything having to change. So an enzyme in Dunaliella is exactly the same as the same enzyme in a non-salt tolerant organism, whereas the enzymes and everything else in Halobacterium are not the same, and they will not act, they will not work in low salt. Okay, so as I mentioned when I first introduced the high salt mechanism, they don't accumulate sodium ions. What they do with the sodium ions is they're used to stabilize the external part of the membrane and the cell wall. There's a sort of an area, particularly in gram-negative type organisms, and some archaea have similar sort of cell walls in the sense that they are, they're quite, some of them are really quite different from sort of normal prokaryotic cells. So sodium ions will actually interact very carefully with the glycoproteins and other parts of the cell wall, and to keep the cell wall sort of intact. Whereas inside the cell, it's not sodium that's accumulated, it's the alternative cation potassium. So to stop all these negative charges, the CO- charges falling apart, then it's actually it's actually potassium that's doing this inside the cell, and it's sodium that's doing it at the level of the cell walls. And we can tell that when you grow Halobacterium in 4.5 molar sodium chloride, which is quite capable of growing in. Then the cells contain about 4 molar potassium chloride and about 0.7 molar sodium chloride. So there's a fair bit of sodium, far more certainly than in the low salt-in mechanism, but potassium is definitely the major cation, even at these very high sodium chloride levels. Now, that line at the bottom of the slide there, they're saying when they're growing at 4.5 molar sodium chloride, we get this and that. That is a very difficult experiment to do. It's not been done all that often. We've done it in my lab many years ago. The problem you have here, if you imagine you've got Halobacterium in a medium, so we take them into a test tube or whatever, we can spin the cells down, we can pour off the supernatant, as you've done many, many times in labs, and you've now got a pellet of cells. The problem is that within the pellet, you've also got some of the medium. So if you think about, you've probably seen this in the lab, if you centrifuge something down a normal bench centrifuge, pour off the supernatant, and then wait a moment, you'll always find some liquid in the bottom. It's really quite difficult to get rid of that last bit of liquid. Now the problem for us in this experiment is that liquid is full of sodium. So if we just then took that pellet with a bit of medium and then measured all the ions, then when this was done initially, they got really high levels of sodium. They got maybe 2 and 3 molar, because this is actually contaminating sodium from outside the cells. So the trick that we use and others have used is you actually put a layer of silicon oil into the bottom of the of a Eppendorf tube. You add your cells to the top of that, and you spin them through the silicon oil. So basically, silicon oil forms a barrier between the medium and the cells. So you will still get a very small amount of medium going through the cells, but much smaller. And if you really want to do this very carefully, you can actually use, we've now used fluorescent probes, and we did it, we actually used radioactive probes to actually work out how much of the volume in the pellet was extracellular, and you can then completely account for the sodium. So I just wanted to give you just that little bit of background. And sometimes we put something up on a slide like this, and it sounds very straightforward. Believe you me, to actually get that result was anything but straightforward. It was really tricky to exclude the external salt from the pellet, so you can then take the pellet, break it down, and then just measure the sodium, potassium, and chloride in the overall pellet, and then you can get your concentrations. But it's been done enough times now that I don't think there's any doubt that it's true. And also, if you actually extract enzymes from Halobacterium and then expose them to high levels of sodium chloride, they are inactive. If you expose them to high levels of potassium chloride, they're perfectly happy. So we can actually show directly that 4 molar sodium chloride does actually sort of kill off, if you like, the enzymes of Halobacterium. Okay, so they're also highly acidic, this CO- group, but these proteins are also contain very low levels of hydrophobic amino acids, and this is something, again, that Dr. Baker will tell you more about. But what it means is it makes them very polar proteins, and so if you're actually going to purify these proteins, you have to take into account that they are really sort of very highly polar, and you need these levels of potassium inside the cell to keep these polar proteins in solution. If you didn't have that level of positive charges, they would literally come out of solution and clearly be denatured. So the number one take-home message then is that high salt is absolutely essential for Halobacteria, and it's essential at a very fundamental level within the cells. You cannot genetically engineer a Halobacterium to become like a Dunaliella. Even in today's level of sophistication, I don't think that would be possible. You'd have to change almost every aspect of the cell and make it, reverse this sort of requirement for the high levels of salt. So this is very much an irreversibly these changes are really irreversible, and it means that you find Halobacterium in salt lakes. You don't find it in rock pools. You never find it in a rock pool. You don't find it anywhere where the salt concentration is going to lower to any significant level. It can increase quite a bit. In fact, Halobacterium can survive sort of salt crystals, as in the salt mines, for example. But if it was lots of rainfall and was diluting the salt lake, then Halobacterium would not be viable. Now, one thing I always do before I give a lecture to any class that I've given a few times, I always look through the literature again and just make sure nothing is dramatically changed. I always sit down the night before and think, does all this make sense? Try to think it from your point of view. It's quite hard with the halophiles because I've worked with them for so long. The only thing that always nags at me when I'm sitting at home looking at these lecture notes is, what about the chlorine ions? You've got all these anions inside these cells. What the devil are these chloride ions doing? And the answer is we don't really know. Again, I've checked this up. We know they control the levels of chlorine, and we know that they are probably compartmentalized to a certain extent within the cells, but it's a prokaryotic cell. It doesn't have vacuoles or anything like that. So I think it's still a bit of a mystery that all of the stuff I've told you in the slides makes sense. But if you read these slides carefully, what I've just said to sort of trigger you, I've not really said anything about chlorine, the anions at all, really. They're there, they don't change. It appears if you get 4 molar chlorine ions in the medium, you've got more or less 4 molar inside the cell. That seems to be the same. Whereas with the low salting mechanism, the chloride ions are definitely moved out with the sodium, so there's no problem there. But with this one, it always just nags at me a little bit as to what exactly are the chlorine ions doing. There are a couple of papers talking about it, but nothing that's really very helpful to explain that. So I'm interested to see whether it's something I've mentioned with Dr. Baker before, so we'll see if anything else has come up. But from my point of view, I'm happy for you to accept that potassium sodium is a key difference. The irreversibility is definitely true, but there's just this little bit about what exactly are the chloride ions doing. Right, so to end this lecture, just a comparison between the two mechanisms. So we've looked at the high salting mechanism relatively briefly today, but as I say, you get more of it from Dr. Baker. I've looked at the low salting mechanism in more detail because that's something that I've worked on myself for quite a bit. And essentially, it's a trade-off, as all these things are. So the high salting mechanism costs much less energy. So it's far less energetically costly to transport potassium into the medium, transport sodium out. So once you've got all these changes to your ribosomes, enzymes, membranes, then the actual day-to-day running, if you like, of a high salting mechanism is quite low. But the problem is there's no flexibility to colonize low salinity habitats. So if high salinity habitat disappears, then Halobacterium has nowhere to go, if you like. If a salt lake was diluted and became much lower salt concentration, then Halobacterium would simply die, whereas the Dunaliella type of, or Halomonas type of low salting could adapt. They would actually adapt to the changing salt concentration, and you'd actually then they'd be able to carry on growing in the lower salinity habitat. But the cost to the Dunaliella Halomonas is in a very high amount of energy, so it's very energetically costly to grow with compatible solutes. It's a large amount of your CO2, a large amount of your carbon source, CO2, or a large amount of energy in form of ATP is actually being used to produce the compatible solutes. Okay, so with each section of these lectures, this is the end of the halophiles. I'll always give you 2 or 3 references. So the first one is one of mine from a very long time ago with a guy, Steve Cummings, who's now a professor of microbiology in his own right and has been for many years. But he did his PhD back in the 1990s, and we were working with Halomonas, and that paper talks about giving it glycine betaine as a carbon and energy source and seeing what the cells does. So that allows you to see a bit more about the control of glycine betaine and ectwine and how Halomonas sort of juggles between the 2 compatible solutes. The DaCosta review is the one that I asked you to look at for the lecture that was missed on Friday. So that's a review of compatible solutes. That talks about a wide range of compatible solutes and really builds on what's in handout 2. And the Schubert et al. One is one where they're looking at finding the bacteria in salt mines, and that they've actually managed to survive for many hundreds of millions of years. So that one's more a general interest one, but it also has some interesting information as well. So what I always urge students to do is to have a look at these references, if possible, as soon as possible. Read them, be sensible how you read them. The Cummings and Gilmore one is a classic research paper. It's all the methods in it. Don't read any of the methods. You're not going to have to do any of the experiments. Read the abstract, the introduction and the conclusions, and then make some notes into your lecture notes. That's the way I would suggest to do it. The same, the Da Costa review, take bits of it that are of interest to the compatible solids you've heard about and annotate your reviews. And the Schubert one, I say, is really just of general interest, hopefully, that you'll be fascinated by the changes, the amount, how these organisms can survive for long periods of time. And again, you can just make a few notes on that. Okay, so that's a really good place to stop for this morning. And so I'll carry on tomorrow with the thermophiles as the next group. Thank you.
I was doing a different module last week.
Oh, sure, yeah, absolutely. There you go. So that's number one. That's number two.
Thank you so much.
And number three. That's number three.
Oh, yeah.
There we go. There you go. All right.
You mentioned all these diseases.